Hello and welcome to our webinar. I'm Gaurav Agarwal, Field Application Specialist at Eurofins DiscoverX. In this webinar, I'll be focusing on our single donor-derived immune effector cells, a part of our innovative killer cytotoxicity assay platform, and will show you how these effector cells can help in successfully implementing your ADCC assays in QC lot release programs for your therapeutic antibody drug candidates. So join me to learn more about our killer CD16 effector cells. First, let's understand what are cytotoxicity assays and why do we need them. Let me start with a bit of background. As we know, in the last 10 years or so, the use of antibody therapy in the field of oncology has shown very promising results. Due to their high specificity, antibodies represent a promising method for targeting a specific molecule with high selectivity. And for that reason, therapeutic monoclonal antibodies are the fastest growing class of biological therapeutics, and more than 50 monoclonal antibodies are already approved by the FDA for clinical use. These therapeutic antibodies can function in a number of ways for the treatment of cancer, such as blocking the interaction of cell surface receptors with the ligand, thereby blocking signaling events that may suppress body's immune surveillance system. But one of the most interesting and effective ways that some antibodies act is by binding to an antigen on the surface of the cancer cells and thereby recruiting body's own immune effector cells, which then engages and kill the cancer cells. Some of the well-known effector cells belong to both innate and adaptive immune system, such as macrophages, cytotoxic T cells, natural killer cells, and monocytes, to name a few. But an efficient development of these complex cancer immunotherapy drugs requires robust cell-based assays that could actually allow researchers to determine whether a particular drug candidate can engage the immune effector cells to detect and kill the target cancer cells. Such assays are termed as cytotoxicity assays. So let's start with understanding how these therapeutic antibodies engage the immune effector cells to target cancer cells. As we know, tumor cells do express specific cell surface antigens, which differentiates them from normal cells in the body. These differentiating antigens are excellent candidates to target these therapeutic antibodies against, thereby letting the normal cells to be spared from their cytotoxicity. Now on the right hand side here, I have drawn a schematic of a tumor cell expressing a tumor specific antigen that can be targeted with a therapeutic antibody. Now when the antibody recognizes this antigen, it binds through its FAB domain, and the other end of the antibody, called the FC region, engages with the immune effector cells. So the question is, how does the immune effector cells bind the FC region of the antibody? Well, that happens through a receptor they express on their surface called the FC gamma R3A, also called as CD16. In fact, there are different FC receptor classes that play an important role in augmenting some of the key signaling events for immune checkpoint receptors. You can learn more about FC receptor engagement in another one of our webinars by Dr. Jean Lemerdine on our website. So to continue, the synapse formed between the tumor target cell and the effector cell through the antibody activates the effector, and based on the effector cell type, the tumor cell will be killed either by enzymatic lysis or cytokine release, and also in some cases by induced apoptosis. So this process, where the tumor cells are targeted and killed by the immune effector cells through the antibody, is referred to as antibody dependent cellular cytotoxicity, or ADCC. Therapeutic antibody drugs whose mechanism of action is through the ADCC process, which mostly belongs to IgG1 isotype, an MOA reflective ADCC assay is necessary to quantify and monitor their effector functions. Thus, The clinical development of these therapeutic antibodies absolutely depends on availability of a robust ADCC assay that can support the QC lot release program for the drug candidate. As I've covered in the last slide, ADCC process is complex and have too many moving parts, so to speak, and it can be challenging to develop an MOA reflective, reproducible and robust ADCC assay. So let's review some of the most desirable characteristics for an ADCC assay to be implemented in a lot release program. Firstly, the assay should be reflective of the mechanism of action of the drug. This is important as that the assay should be reflective and not predictive of the true mechanism of action of the drug molecule, or else the assay would be considered as a surrogate assay. In the end, FDA is looking for a MOA reflective ADCC assay, thus it is important to select the assay carefully. Second, the assay should produce repeatable, robust, precise and accurate results. These are the key hallmarks for an assay to be implemented in a QC lot release program, where it is essential to obtain coherent datasets to demonstrate drug product identity, purity, stability and potency. The third is the availability of effector cells themselves with minimum donor-to-donor variability as well as lot-to-lot variability. This is of critical importance since the effector cells are known to introduce significant variability in cytotoxicity assays. But we have good news. Our killer cytotoxicity platform offers solutions to all the above concerns and support your lot release program. Firstly, our killer cytotoxicity assays are absolutely reflective of the drug MOA and specifically measures target cell death, which also is the assay endpoint. Our killer platform presents an innovative approach to accelerate the development of your cancer immunotherapy drugs, including antibody-based therapeutics and cellular therapies like CAR-T. Next, killer assays produce highly repeatable, robust and accurate assays. These are not radioactive or fluorescent dye-based assay systems and thus are not leaky, which is one of the main issues with the several other cytotoxicity assay systems. As you will see in next slide, that the killer assays are based on a reporter protein which is stably expressed in the killer target cells and produces a signal which is only from the target cell death and will not account for any effector cell death if that is happening in the assay. So the signal is extremely specific only to the target cell death. To learn more about our simple, non-radioactive, scalable and dye-free killer cytotoxicity assays, please join my other webinar available on our website. Now, addressing the third concern regarding the effector cells associated variability is the subject of this webinar. In the coming slides, I will talk about our killer effector cells and show how they produce robust and reproducible cytotoxicity assays, making them well suited for discovery, characterization, and more importantly, lot release programs. So let's begin with reviewing some of the key challenges with the currently available effector cells. Mostly, it is the primary cells that are used in ADCC assays as effector cells. These are mainly PBMCs or natural killer cells, isolated from the primary cells from individual donors. But there are some big drawbacks using these primary cells in cytotoxicity assays. Firstly, it is the donor-to-donor variability itself. What it means is that cytotoxicity assays shows higher variability between experiments when the effector cells that are used are derived from different donors. So this is by far the most prominent issue with using primary cells in these assays. The second is to be able to get enough of these primary cells from one single draw from an individual donor. And there is always a limited number of cells that can be isolated from one draw. Lastly, the effector cells isolated from primary cells often produces low assay windows in ADCC assays. In other words, the killing activity of these effector cells is not very potent. Hence, even a very effective antibody like rituximab, which is an anti-CD20 antibody approved by the FDA, for the treatment of lymphoma is not able to produce a very high uh, uh, killing potential dataset. The data on the right here shows various ADCC assays with PBMCs using rituximab. As evident, PBMC from all these different donors produce less than 30% ADCC activity, which means less than 30% of the cells in the assay are being killed by the effector cells from any of these donors. Next are the NK92 cells, which are expressing CD16 on their surface. These are cell lines used by many researchers for their ADCC assays. But the cells have a different set of drawbacks, but mainly these cells are quite challenging to grow and passage. Secondly, these cells have a very high non-specific killing activity, which makes them incompatible with different backgrounds, as they induce a very high non-specific killing activity, which reduces the assay window significantly. ARH77 cell background is one of the examples where these NK92 cells result in almost 60 to 70% antibody independent non-specific killing. Another significant drawback with these NK92 cell lines is their exorbitant licensing fees that prevent many research programs in even accessing these cells. Now, the other option that's currently available are the effector cells that are engineered to express an NFAT reporter system. In addition, they also stably express CD16 receptor. When used in ADCC assays, the CD16 receptor engineered in these cells engages with the FC region of the antibody to bind the target cells, which activates a signaling cascade resulting in the activation of a luciferase gene transcription. So the ADCC assays driven by these effector cells basically measure a signaling activity but do not measure any actual cell death of the target cells. As we discussed, the MOA of the drug candidate is to engage the effector cell to actually kill the target cells. So these assays are not reflective of the MOA, but only at best are predictive. Hence, they yield a surrogate assay that at later point needs to be supported by an MOA reflective assay. So it is important that that you choose an ADCC assay carefully based on all these concerns. And as I mentioned, all the gaps here we just talked about could be addressed by our CD16 effector cells. And let's move forward and let me give you a quick overview comparing our killer CD16 effector cells with these primary cells. In the schematic on the left here, I have shown an effector cell such as a PBMC or an NK cell found in the body. These cells endogenously express a CD16 receptor on their surface that engages with the FC region of the antibody, as we discussed earlier. Upon engagement with the antibody through the CD16 receptor, these cells are activated and release enzymes like perforins and granzymes that kill the target tumor cells. Now, although ADCC assays driven by these cells are reflective of the MOA, they introduce significant challenges, as we just discussed in the previous slide. Now let's compare these cells with CD16 effector cells. Now CD16 effector cells are single donor derived cytotoxic T cells which are engineered to stably express CD16 receptor. As you can see, just like the PBMCs or NK cells, they engage with the target cells through the CD16 receptor on their surface. Since these are cytotoxic T cells, they release cytokines to kill the target cells and provide a true MOA of the ADCC assay, which is target cell death. In addition, the killer effector cells also addresses the concerns of the primary effector cells we just discussed by eliminating the donor-to-donor variability as they are derived from one single donor which was drawn at one single occasion. They also provide very low background killing and results in higher assay windows. The other advantage, them being cytotoxic T cells, they have a rapid killing kinetics as well as more potent and are more potent in producing a higher killing percentage in ADCC assays. Let me add a few other technical details to these CD16 effector cells. As I mentioned, firstly, these are human cytotoxic primary T cells engineered to stably express the V158 variant of the CD16 receptor. This V158 variant is known to have higher affinity for IgG. They are manufactured following a stringent SOP in a quality environment, ensuring uniformity in manufacturing and low lot-to-lot variability, and produces low background with higher killing capacity, ideal for an ADCC assay which is implemented in a QC lot release program. Let's look at some of the key cell surface markers on these cells. Firstly, since these are cytotoxic T cells, they are CD8 positive and CD4 negative. The FACS data here shows over 99% of cells are CD8 positive. Next, they are also CD3 positive. CD3 is a hallmark of cytotoxic T cells. Now, them being CD3 positive, they can also drive T cell redirection applications, which I'll be discussing in the later part of this webinar. Lastly, these cells are engineered, as I mentioned, to express CD16 receptor on their surface, and from this data, almost 100% of cells are positive for CD16 expression. In a nutshell, these are CD8 positive, CD3 positive, and CD16 positive cytotoxic T cells. Next, let me discuss a little bit on how our killer cytotoxicity assays work that we use to evaluate the performance of these effector cells. So like most of our cell-based assays, killer platform is also based on our enzyme fragment complementation technology, or EFC. In short, EFC is based on split beta-galactosidase system, where the enzyme is split into inactive fragments called ED, or the enzyme donor, and the EA, or the enzyme acceptor. When these two fragments are put together, they complement each other to form a functional beta-gal enzyme. We add a chemiluminescent substrate in the assay that this active functional enzyme hydrolyzes to produce a light signal that can be read with any benchtop luminometer. Now the question is how we use this EFC technology to set up killer assays. In fact, it is a very simple assay design depicted here. The ED fragment of the beta-galactosidase system, so enzyme is tagged to a housekeeping gene, which is stably expressed in a target cell background. When these target cells are lysed in an ADCC assay, the reporter protein is released from the cells where it complements the enzyme acceptor fragment, and in presence of the chemiluminescent substrate produces a quantitative light signal. So here is an assay outline on how killer ADCC assays are set up with the CD16 effector cells. To start with, you can choose the cell background of your choice. We have over 40 ready-to-use cell backgrounds available in our catalog that you can pick from. Good chances that you will find your desired background, but if not, we have a custom assay development team who can build one for you. We also have available do-it-yourself kits if you would like to build the cell lines, the killer cell lines in your own labs. Next, you will add your drug candidate in the assay, incubate for 1 to 6 hours, followed by addition of the CD16 effector cells. Good thing about these effector cells is that they can be used with other ADCC assay systems where MOA is to measure target cell death. So they are not restricted to be used just with our killer cytotoxicity assay platform. Now upon the addition of the effector cells, the antibody will form a synapse between the effector cell and the target cell that activates the effector cell to kill the target. Upon lysis, the killer reporter protein is released from these target cells to combine with the enzyme acceptor fragment added alongside the beta-gal substrate. Now the complementation of these two inactive beta-gal enzyme happens in the medium, and this active enzyme hydrolyzes the chemiluminescent substrate to produce a quantitative light signal as I described in the previous slide. So this is a simple outline on how to set up ADCC assays using CD16 killer effector cells. Moving forward, let's look at some of the ADCC data with these killer effector cells. Here are two data sets. On the left, we have the rituximab model with ARH77 cell background, and on the right, the trastuzumab model with SCOV3 background. Let's focus on the data set on the left at first. Here the blue curve is with the killer CD16 effector cells versus PBMCs in red. What we typically use for PBMCs is a ratio of 25:1 effector to target cell ratio, which we have already reduced to 10:1 for killer CD16 effector cells. Yet see roughly a 5-fold increase in signal to background, while 2-fold increase in the max cytotoxicity relative to PBMCs, while the change in EC50 is quite small, thus keeping the pharmacology consistent. Now on the right hand side, we see a more difficult to kill cell model, SCOV3. Here what we observed was that the PBMCs produces a much shallow response with less than 3-fold assay window, but we saw a much better response with CD16 effector cells with over 40-fold assay window. So these completely changed the assay. These data set demonstrates a highly robust ADCC assay from the killer CD16 effector cells versus PBMCs. We have seen some of our customers reviving their projects using these killer CD16 effector cells for their drug candidates that they have set aside just because they were not able to obtain a reasonable assay window with PBMCs. Next, also an important data set here comparing the lot-to-lot reproducibility with the killer CD16 effector cells, as it is one of the key consideration in quality environment. In this rituximab model with ARH77 cells, 4 different lots manufactured over a course of 2 years were analyzed, and you will see that the lot-to-lot variability is much lower, around 35%, versus the donor-to-donor variability with PBMCs, which could be nearly 60% as seen in this experiment. Moving forward, we have another data set which is critical when you are considering or selecting critical components for an assay to implement in QC lot release programs, is looking at analyst to analyst variability. In this study, we included 2 analysts who thought 2 different vials of the effector cells and performed the ADCC assays side by side. The data demonstrate a very low inter-user variability with less than 20% CVs, making these effector cells highly amenable to be implemented in labs across several sites of your organization or to your CRO partners. Now I will switch gears a little bit and talk about other potential applications for killer CD16 effector cells. One of the other key applications for these effector cells would be in T-cell redirection. A little bit of background on T-cell redirection. Redirection of T-cells to target tumor cells holds a good promise for treatment of cancer. There are various types of T-cell redirection therapies in development, and one of the prominent approach is to use the bispecific T-cell engagers or BiTEs. These are dual specificity chimeric antibodies designed to harness the power of immune effector cells to target tumor cells. Blinatumomab is one of the successful BiTE molecules approved by the FDA for the treatment of lymphoblastic leukemia. As shown on the schematic on the right, one arm of this BiTE molecule binds to CD3, an antigen found on the surface of T cells. Now, more commonly, researchers use pan T cells, which are isolated using negative selection for T cell redirection applications. Now, while the other arm of this BiTE molecule is engineered to bind a tumor-associated antigen such as CD19. Interestingly, the killer effector cells are in fact cytotoxic T cells and endogenously expresses high levels of CD3, and therefore they are amenable to be used for T-cell redirection application. Here is the FACS data that I showed you earlier for CD3 positive staining, and as you can see, there are very high percent of cells which are positive for CD3. Let's look at some of the data set with T-cell redirection application. So we evaluated the T-cell redirection with blinatumomab using killer cytotoxicity assays, Initially with pan T cells, the data on the left is with pan T cells. These cells are isolated by negative selection, as I mentioned, and we perform the redirection application using our killer Raji cells. Now, Raji cells are endogenously expressing CD19 on their surface as they are derived from B lymphocytes. As you can see that in absence of any effector cells, we have a very low cytotoxicity as shown by the teal dots. But a 24-hour incubation with blinatumomab followed by the addition of pan T cells produces a very good dose response curve. So if you compare that to the killer effector cells on the right side, what you can see from the time course there, that the kinetics is much faster than we typically see with pan T cells. An optimal response that is comparable to what we saw with pan T cells could be achieved with only 6-hour incubation of the BITE molecule with the killer CD16 effector cells. Also importantly, the EC50 of blinatumomab as reported in the BLA is between 10 and 100 picogram per ml, and the above assay formats, both of them produced EC50s within this range, thus keeping the pharmacology consistent. So to conclude, our killer CD16 effector cells are capable to drive robust and reproducible effector cell performance, not only for ADCC, but also for T-cell redirection applications. Let's review some of the key features we learned today about killer CD16 effector cells. Firstly, these are single donor derived primary human effector cells. So the benefit in using these is obviously eliminating any donor to donor variability in the ADCC assays to obtain data with high reproducibility. Secondly, these are effector cells that are primary cells engineered to express CD16, which is required for mediating ADCC and thus keeps the MOA of the assay consistent. Next, we have observed high lot-to-lot reproducibility, which will enable a long-term support for your QC lot release programs. Next, these cells drive target cell killing in ADCC and T-cell redirection applications. Thus, they reflect the true mechanism of action and fulfills one of the key requirements for an assay to be acceptable by the FDA. And also, killer CD16 effector cells produces a significantly higher signal to background ratios and thus provide an improved performance compared to PBMCs, thus enabling their use in QC lot release programs. With that, I would like to thank you for your attention. Please visit our website for more information on our killer CD16 effector cells or contact our technical support team if you have any questions. We'll be very happy to help. Thank you.